So, thanks for joining us today. I'm going to introduce you to our speaker today, who is Josh Arnold. Josh is the Aquatic Specialist here at LGROW, and he is conducting his graduate research on freshwater mussels in the watershed. So we are happy to have him, and he is going to be sharing some of what he knows about mussels, why they're important to the watershed, and some of the research that he's doing on them. So this will be about a half hour, maybe 40 minutes long. So we'll get you out of here a little bit early, and hopefully you'll learn something. Quick fact, if you are joining us on Zoom, go ahead and throughout, if you have questions, feel free to type them in the chat. And if you're joining us on Facebook Live, we're so excited that you're seeing us on our page, and feel free to comment on the video with questions, and we will make sure that Josh addresses those for you. So with that, I'm going to turn my video off, and I'm going to turn it over to you, Josh, to present your webinar to us.
Hello, everybody. Thanks for joining us today. Once again, my name is Josh Arnold. I am the Aquatic Specialist at LGROW, and I'm also an aquatic biologist grad student at Grand Valley State University. And today I'll be talking to you about the ecology and life history of the freshwater mussels of North America, with an emphasis on mussels here in Michigan, as well as in the Grand River. So as some quick introduction, there's roughly 1,000 species of freshwater bivalves around the globe. And about 700 of these belong to the order Unionidae, which we will henceforth refer to as freshwater mussels. As you may not have known, North America is considered to be the biodiversity hotspot of the world for freshwater mussels. There are roughly 300 native species. This number is always changing due to new genetics work. To put it in perspective, Europe only has 11 native species of freshwater mussels. This is just due to pure luck and the geography and geology of North America compared to Europe. Here in North America, most of our rivers, or major rivers at least, run north to south, while in Europe they run east to west. And due to glaciation covering up parts of both continents, the rivers that run east to west will take millennia to be re-musseled, while in North America, they can be repopulated much faster due to the north and south direction of the streams. This diversity is not spread evenly throughout the states. Alabama has roughly 178 species of mussels in comparison to 450 different species of fish, while the Amazon River drainage, which is the most speciose drainage for freshwater fish, has over 5,500 species of fish, but it only has 38 species of freshwater mussels. Even though we have the most amount of freshwater mussels in the world, they are also the most imperiled group of animals in North America. Roughly 75% of all North American mussels are currently listed at either the state or federal level. This could includes species of concern, threatened, endangered, extirpated, or even extinct. In 1993, 21 species were already considered extinct. This number is probably closer to 30 or 40 nowadays. And mussels had the highest extinction rate of all North American freshwater groups. In 1999, this rate was 1.5% species per decade, or like 3 to 4 species every 10 years. And this number is predicted to reach upwards of 6 or 7, or 30 to 40 species per decade. As we can see here in the chart on the right, only about 24% of the mussels are considered stable, while the rest are either extinct or now threatened with extinction. But here in Michigan, we have 46 species of mussels. And once again, less than 1/4 of our mussels are considered secure, while about 10% of our mussels are already extinct in the state, and about 1/3 are considered endangered. And this includes 4 federally endangered species. Here in the Grand River Watershed, though, we're a little ahead of the trend. About half of our mussels are considered secure, and only 1/5 of our mussels are considered to be state endangered, with 1 federally endangered species found in this watershed, the snuffbox mussel, which I'll talk about more in greater detail later. But why should you care about freshwater mussels? Freshwater mussels provide many ecosystem services and are considered to be ecosystem engineers. Depending on the mussel and how big it is, they can filter more than 1 gallon of water per day, greatly increasing water clarity. As you can see in the picture on the right, after 24 hours, that tank is completely clear of water, of sediment, while the tank on the left is still full and chocolatey. This can allow more light to filter in through the water column, allowing for more aquatic vegetation to grow, to provide better habitat for fish that we all like to fish for. Due to all this filtering, they provide an important link between the water column and benthic community through their poop or their pseudofeces, which is mucus-bound packs of stuff they didn't want to eat. That helps to provide nutrients to the benthic community, where around mussels, there's higher levels of periphyton, which is aquatic algae, as well as aquatic insects. We might have a question. In addition, they're also an important food source for many species, including muskrat, river otter, heron, freshwater drum, just to name a few. But for the past 150 years, mussels populations have gone down a lot. This can be due to overexploitation in the late 1800s and early 1900s. Freshwater mussels, like any organism that has a nacreous shell, can produce pearls. However, marketable pearls at least, only occur in a small percentage of mussels. They did the math, and it's roughly 1 mussel will produce a marketable pearl for every 10,000 mussels. At times in the early 1900s, freshwater pearls were the second highest profit of natural industries behind timber. And then starting in the early 1900s, freshwater mussels were being used more and more to make buttons. In 1916, almost 6 billion mussel buttons were produced. And this comes at the expense of millions, if not billions, of freshwater mussels. These pictures are a little blurry because they're a century old, but you can see just piles and piles of mussel shells. I always equate this to those old pictures we saw of American bison skulls, but instead of hundreds, if not thousands, this is hundreds of thousands, if not millions of individuals that were collected and killed for either pearls or buttons. And then starting in the mid-1900s, the United States went through a wide-scale habitat alterations. This includes dams, which greatly alter the water quality, as well as substrate above and below the dams due to increased sedimentation, increased temperatures, varying flow regimes. They also prevent fish passage, and I'll talk about later how fish are important to mussels. In the United States alone, there are roughly 75,000 dams over the height of 1 meter. To put that into perspective, that is 1 dam per day since Thomas Jefferson was in office. And there are roughly 210 dams over 1 meter in the Grand River watershed alone. And also, we channelized a lot of our streams in the mid-1900s. This also alters the stream characteristics, increased flooding, higher erosions, higher temperatures, less habitat for mussels and fish. And there are roughly 200,000 miles of streams that have been channelized in the United States. To put that in perspective, there's only 36,000 miles of streams in Michigan. So that'd be channelizing each stream in Michigan 8 times over. And then finally, in the late 1900s and currently into the 2000s, invasive species have wreaked the toll on the freshwater mussels. Starting in the late 1990s was the zebra and quagga mussels, referred to as Dresden mussels, not a species of freshwater mussels. They originally come from Eurasia, namely the Caspian Sea. They were brought over in large ocean-going cargo vessels that dumped out their bilge water in the Great Lakes, and they can reach really high densities, such as over 5,000 individuals per square meter. And in the upper right-hand corner, you can see what they do to mussels. They basically completely cover our native mussels and outcompete them and make it very costly for the mussels to open up, and that they basically starve them. And then a little bit more recently, the round goby, also native to the Caspian Sea area, was once again brought over by oceanic cargo ships. And these actually outcompete the native benthic fish that a lot of these mussels rely on for reproduction. In addition, they can also eat native mussels, but they kind of prefer to eat the zebra mussels and quagga mussels, but nowhere near in a high enough densities that would make a difference. Then common carp, Asian clam, black carp, are all invasives that are also wreaking a toll on our native mussels, but they're a lot less studied, so we don't know as much on how they're affecting the native mussels. And then now into perhaps the most interesting part is the ecology and life history. In the upper left-hand corner, we can see just the size difference between two adult individuals. The one on the bottom is an adult female rayed bean, which is max size about 1.5 inches, while the one above it is a Wabash mussel from Kentucky, which will get about 16 or 18 inches long. In the bottom left, we can see a Lampsilis mussel that has a mantellore, and I'll get into more details about that in a bit. Mussels are pretty much found in every water body in the entire United States. They can be found from headwaters to the largest lakes, including the Great Lakes. This includes one tidal species, which can tolerate brackish water for certain times. Most species will prefer the gravel or cobble substrates. It makes it easier for them to bury into the substrate in the winter to avoid being frozen. But between the extremes of constantly shifting sands and bedrock, they can be found in pretty much every type of bottom. And they're extremely sensitive to sedimentation. If the sedimentation rates are so extreme, these mussels that are experiencing sedimentation will effectively be buried alive. For their diet, as I previously mentioned, mussels are filter feeders. All mussels have two siphons, as you can see in the left-hand picture. The one on the right is the in-current siphon. They pull water into their gills by expanding their muscles in their body. And then that water is filtered through their gills and then pumped out through the out-current siphon, which is on the left. The young mussels can also do pedal feeding. They have a muscle that sticks out of their body, which is called the foot, and they use that to root around in the substrate to get various nutrients. Mussels mostly eat plankton, bacteria, and detritus, but they'll eat a lot of, as long as it's pretty small, they can filter it. But if they get stuff that they don't like, such as pieces of dirt or tiny rocks in them, they can bind these up in mucus and exude them from the body, and that is called pseudofeces, which can help then give nutrients to the surrounding area. Now, getting into what is probably the most interesting part about freshwater mussels is their reproductive cycle. Freshwater mussels are sexual organisms. They reproduce sexually. Some are hermaphroditic, but still prefer to produce sexually versus asexually. Male mussels will expel sperm into the water column upstream, and female mussels will intake that sperm downstream. The sperm then fertilizes their eggs, which develop in their modified gills called marsupium. These fertilized eggs then develop into larval mussels, which we call glaucidia. In order for glaucidia to develop into juveniles, they must first infect a host. In most cases, this host is a fish. I'll get into a bit on how they do that. Once they get attached to the host, either on the gills, fins, or skin, they spend a certain amount of time, a couple weeks to a couple months on the fish, and then eventually drop off and land the substrate and spend the rest of their adult lives in the substrate, not moving much. So for host selection, as I mentioned, all mussels except for one use the fish as their host. This odd one out is the salamander mussel, which actually uses the common mud puppy as their host. We have this species in, might be a question. Yes, we can give you a recording of this afterwards. The salamander mussel uses the common mud puppy, which is found here in Michigan, but not in the Grand River. It's only located in the southeast part of the state. And then there are host generalists versus host specialists. Host generalists are species of mussels that can use a couple to dozens of different species of fish as their host, while host specialists only use one or two different fish species as their host. And the picture on the right here, we can see a zoomed in picture of a glaucidia attached to a gill filament of a mussel. They feed on both the blood and the immune response, which is what's surrounding the Glaucidia. That's how they survive and develop. So to get into some of their infestation strategies, broadcast spawning is probably the most ancient and basic one. Basically, they expel their young Glaucidia into the water column. Just millions upon millions of very tiny Glaucidia. An example of this would be the eastern pearl shell Atlantic salmon. This is thought to be an effective strategy for when the host species is in high densities, such as species of fish that make migrations, such as Atlantic salmon on the East Coast, as well as like shad, and on the West Coast would be your Pacific salmons, your kings, your Chinooks, and that kind of stuff. There's also species of mussels that will expel their Glaucidia into the substrate, and this is to target species of fish that filter through the substrate, such as our sucker species, as well as freshwater drum. And then conglutinates, or mucus-bound packs of Glaucidia. They can be shaped like eggs or macroinvertebrates, and their hosts are often small fish that feed on insects. In the picture on the left here, we can see a type of darter from down in Missouri. Then right above its head are two little, almost look like insect larvae. Those are actually the mucus-bound packs of Glaucidia. And once the fish would bite into those, that thin mucus layer breaks down and they get infected on their gills. And then getting into more of the large, flashy infestation rates would be your mantellores. The mantle is part of the mussel's body. Females have a modified mantle that resembles a prey species. These can be large lures, such as the one pictured on the left, which target top predators, such as your bass, gar, and pike. And there are species that have smaller lures for smaller species, such as the bluegill and other panfish. To do a brief description of how this strategy works, the mussel will flap this lure, which looks surprisingly like a fish, even has an eye spot and a lateral line in the water column to attract a fish. This white thing that you can kind of see is the marsupium, or the gills, where they keep their Glaucidia. And a lot of the top predators, such as bass, feed through what is called buccal feeding. When they open their mouth very rapidly, which creates a vacuum, which actually ruptures the thin layer of the marsupium, pulling the Glaucidia right into their mouth and onto their gills. And now we have a little bit of a video to show you this in action.
In the streams of Missouri lives the Lampsilus mussel, a simple animal with an extraordinary life cycle. To reach adulthood, its young must spend part of their lives inside a fish, the largemouth bass. To get there, the mussels must make physical contact, a difficult task, as mussels don't swim. But the bass has a weakness. It's a voracious predator of small fish, particularly darters. Even the slightest wriggle of a darter's tail will attract bass. Believe it or not, the fish on the mussel is an imitation, a perfect replica that will lure bass within striking range. The mussel can somehow sense approaching fish and wriggles its lure faster to entice them. If it gets the twitching just right, the remarkable likeness should do the rest. On impact, the mussel squirts its young into the bass's mouth. These snap shut on the gills, like spring-loaded traps. Here they stay, drawing blood from the fish, until several weeks later they drop off as tiny, fully formed mussels. Also a favorite prey of the bass are these striped shiners, and some mussels mimic them Considering mussels are blind and have never seen a shiner, the likeness is incredible.
And then, as I mentioned in that video, it is amazing how close those lures get to the actual fish they're trying to mimic for something that has never ever seen a fish in its life because they don't have eyes. And then moving on to perhaps one of the strangest infestation strategies is host capture. Members of the genus Epioblasma, which includes the snuffbox, which is found in Grand River and in the downtown stretch of Grand Rapids, they actually will lure their prey into their body and then close around its face. This is often done to various species of darters and minnows, and many times, like the picture on the right, which is a snuff box, they have modified shells to hold onto the fish, and these are called denticles or teeth-like protrusions. And these species are often host specialists, and many of these species belonging to this genus are federally endangered. We have a quick video showing this. And this video has no sound, unfortunately. And in case you're curious, that fish is fine. These two species evolved together so that the log perch actually has a thicker skull than most fish to survive that clamping. But unfortunately, just due to the strength that the snuff box can close with, a lot of times when non-target species accidentally trigger that clamping mechanism, they are crushed to death. And finally, our last topic will be mussel conservation. Since they are the most important group of animals, what can we do to help conserve the remaining mussel populations? The biggest thing when it comes to conserving mussel populations is habitat. This can be done by either the removal of dams, such as the picture of the 6th Street Dam here, which is slated to be removed eventually in the future, and this, as well as the regulation of tidewaters from larger dams. And there are more and more habitat improvement projects that are focusing on mussels, such as the Grand River Restoration Project happening in downtown Grand Rapids. The snuffbox mussel is one of the target species to benefit from this project, instead of mussels being more of a byproduct from their benefits. Then what scientists do is a lot of surveys. Surveys are important to determine distribution, as they are a pretty cryptic species. They're actually rather hard to find in some cases. So it takes a lot of effort for scientists to determine where mussels are and to track their population trends and determine if they are either extinct or not. As I said, this tracks current populations, and it also helps to update old data. A lot of times in a lot of states, the data which they base their work off of is decades, if not closer to a century old. And so this older data needs to be updated to see if the mussels are actually still there or not. Water quality improvements also greatly help mussels. Some species are capable of existing at undetectable levels. And by undetectable, I mean some species can exist and live at 1 individual per 100 square meters, while other species will quickly die off if they drop below 10 individuals per square meter. But also, as water quality degrades in some streams, these mussels will actually move up into the tributaries where there's better water quality and hide. And then once water quality improves due to various restoration projects or fixing what might have been affecting the river, they suddenly reappear after what scientists have thought caused them to go extinct in that stretch of the river. And finally, translocation. Translocation is a common practice to save populations of mussels from extirpation, usually due to human effects such as bridge removal or repair, dam removal or repair, or anything that requires heavy machinery in the stream bed. However, it's estimated that only 50% of mussels will survive the translocation process. This is due to stress of being taken out of the water and handled, as well as stochastic effects, which are random events such as flooding or droughts. And this is actually what I study. I study the translocation process in order to better understand how it affects the mussels and to hopefully better improve the process in Michigan so more mussels will survive the process, so we have more mussels to put back in places where they are no longer found. And with that, that's the end of my talk. And if you have any questions, please feel free to either speak up or put them in the chat. And once again, I'd like to thank you all for coming and attending this mussel webinar as part of our LGROW Spring Forum. Thank you.
Thank you so much, Josh. It was great to hear that. Does anybody have any questions about mussels at this point before we let you all go? You can drop them in the chat or in the comments on Facebook. We're happy to answer any questions you might have about mussels. Josh, maybe while we're waiting for some questions, would you be willing to share a little bit about your research that you're currently doing on mussels in the Grand River Watershed?
Yep. My work actually takes place in one of the main tributaries to the Grand River, the Thornapple Watershed, and specifically Cedar Creek. So I conducted my own translocation effort where I took a couple hundred individuals from one site and moved them to different sites with varying differences in habitat and water quality to see not only how does translocation affect the overall health of the mussel, and we measure the changes in length as well as mass and like mortality, but we also want to see how differences in habitat and water quality can affect the overall success, such as even further reduced growth rates or even higher mortality rates. Our preliminary data shows that we want in the future to have translocation efforts target areas that have similar habitat from the source site to put their mussels, because our site with pretty different habitat is experiencing rather high mortality rates.
Thanks, Josh. Anybody else have any questions? Comments? All right. Well, oh, there's one. Josh, is there a place where people can contribute photos or observations of mussels that they have found? Is there any kind of like a citizen science or someplace they should share if they find anything?
iNaturalist is a great place to do it. Either you can identify yourself or the experts on the app can help you identify the mussels you found in your photos.
Yeah, I will vouch for that. I've submitted photos to iNaturalist of mussels before and they have been identified, which was great. What about what's going on downtown, Josh? So can you talk a little bit about the snuffbox mussel and the restoration of the Grand River downtown and how those are related?
Yeah, so the restoration project is to improve habitat for a couple species, including the snuffbox mussel, as well as like lake sturgeon. So before we can do any of the work, such as removing the dams or improving the habitat and installing all those structures that is planned to install, first people will have to go in and actually collect all the mussels to move them someplace else so they aren't accidentally killed in the construction process. So that's why we are moving all these mussels, and that's why my work is important to this process to better understand how to conserve what populations are left. Do you have any, what is the most common mussel in the Grand River or Thornapple? It's hard to say in that general of a term. It varies greatly based off what size stream you're in. Like where my research done is mostly in Cedar Creek, and the most common species is spike, which is pretty common. But if you move down to the Grand River, especially where it's bigger towards Grand Rapids or below that, or in the thornapple, you get more of your larger species such as white heel splitter or three ridge.
I would say from my limited knowledge of mussels that the species that I see most frequently in the Grand, and I live kind of near the confluence of the Grand and the Thornapple, is I see the three ridge all the time. I don't know if that means it's the most common, but it is certainly a very common one.
And it's a rather easy species to identify. As its name says, they usually have three ridges on, and it's pretty unique.
What about other species of mussels, Josh? What do you know about the green mussel?
I don't know anything about the green mussel. They do not exist in Michigan or in any other states I used to work in. So unfortunately, I don't know anything about those.
Great. Good questions, everybody. Any other questions for Josh? All right. If there are no other questions, I'll drag this out a little bit slowly so you can type them in if you're still working on it. We will, as I mentioned, the webinar is recorded, and we will post a recording of the webinar on our LGROW YouTube channel, and we will share that link with those of you who were registered for the webinar and watching via Zoom, and we will also share it in the Facebook event. So if you found us that way or on our Facebook page, you'll be able to go back and watch it again later.
Also, to answer your question, Joyce, because the green mussel is actually a species of saltwater mussel and not a freshwater mussel. Apparently, it's a pretty important one for food source. Speaking of food source, I do not recommend eating any freshwater mussels. A, it's illegal, and B, they taste bad and will make you sick.
Josh, what if you find a big old pile of shells lying next to the river? What does that mean? Does that mean that there was like a big mussel die-off, or is it something we should be concerned about?
Usually you find a big pile of mussels, either in the shallows or next to the stream. That's what we call a middens pile. And that is usually due to either like a muskrat or a river otter. They usually sit in the same spot while they eat their mussels. But that's a really cool place to go and just see all the different types of shells, because they are way better at finding mussels than we are.
Great. Josh, last question. What's your favorite freshwater mussel?
My favorite freshwater mussel is the northern riffle shell, which is a federally endangered species, also a member of the Epioblasma genus. So it's the host capture type, and it is found in southeast Michigan in like the Detroit and the St. Clair River.
Cool. All right, well, thank you again, everybody, for joining us for this freshwater mussel webinar for the LGROW Spring Forum. As I mentioned earlier, we've got lots of other events going on over the next few days up through June 20th. Many of them still have registration open, so we would love to see you at more events. We thank you for coming today, and we will get you the link to this webinar soon. Thanks so much, everybody. Have a great afternoon.